research questions that cannot be answered using traditional mammalian models. So the title of Jan's talk today is Make Do and Make New, Fast-Tracking Neural Repair Using Precursor Neurons. So I'll hand it over to you, Jan. Thank you for joining us today.
Thank you very much for the nice introduction, and also let me know if you don't see me or hear me well. I just had to do a very quick swap over from my office because they just happened today to change cladding just in front of the office, and are grinding off of the cladding. So that was a bit unfortunate, but I hope it all will work out. I'll also start sharing screen and we'll see if that works out as well. And I hope you all also can see and my shared screen here now. Yeah.
Yes, we can.
That's excellent. Anyway, I would also, of course, like to thank Kelly, Michael, and Julian also for giving me the opportunity to present the work for you today here. And what I'm going to talk about is more of a kind of a new concept of an alternative route also to fast repair in the nervous system, both in brain and spinal cord, that we kind of discovered using the zebrafish model. And it really explains how zebrafish can do what they have to rapidly repair, and then also how they can then engage other mechanisms for full tissue healing. And as a very short introduction, most of you know that traumatic injury, different degenerative diseases to our central nervous system is a big problem for us humans, because we can't really repair well. And it's also something that has been studied since quite a long time ago. So the first citation of this, at least that I found, is basically from what is called the Edwin Smythe papyrus that was found in Egypt, and that's more than 3,600 years old. And in that papyrus, they actually describe different injuries and also potentially diseases to the nervous system and basically their outcomes. Since that, we researchers have really done a good job in finding new mechanisms, trying to understand what happens when there is a disease or an injury to our brains, spinal cords, or retinas. And you can see the very rapid increase in basically publications in PubMed that I have plotted here. And today there's about roughly 160,000 studies about brain injuries, and half of them would also contain at least, or mention at least some kind of a way also to cure a damage or disease in the brain. And similarly, about half of the amount of similar studies has been done in spinal cord. Which means that on a yearly basis, currently we more or less publish collectively about 10,000 papers on this topic per year. That's a quite significant amount. The sad thing is, though, that not too much in general has happened since these 3,600 years, because in that papyrus, when they basically say that when you have an injury to the back of the head or to the spine and it's broken, then you have a disease that one cannot treat. And not really much has happened. Of course, we are much better in taking care of patients and in acute medicine and so on. But still, we are really struggling to find very meaningful ways to repair an injured brain from a stroke, from a traffic accident, or any of the many degenerative diseases that are also quite prevalent. So it's clear that we need to understand much more about the processes and all the little tricks we can do to actually help the nervous system heal. And one way of doing that, and that my lab has been working, is to look at it from the other side, in animals that actually can repair their brain and spinal cord and also eyes very well. So zebrafish is one of those. And here's just one example of one of the studies where I tried to actually see how much they can repair. So what you can see here is of course, a zebrafish and a zebrafish brain. You can see to to the right, basically a cross section to the cerebellum. And you can also see that there's a significant damage there. It's basically a surgical removal of about 25% of the hemisphere. Then 1 month later, there to the bottom left, you can see also that the morphology has improved, and you also see a lot of green spots. That's basically cells that were taken up BRDU shortly after injury and would indicate that they were produced and from proliferating cells, essentially neural stem cells, and that essentially new neurons have been made after the injury to repair the tissue. And then also one can look at behaviors and correlate basically the anatomical, the neural regeneration with behavior. You see a graph to the right there, and there one can see basically that they also recover on tissue and functional level relatively quickly. So you see drastic improvements after 2 weeks, and then they gradually improve over the next weeks and months. Morphology will never be completely restored in this very massive lesion, but a lot of cells will recover. And why the morphology doesn't restore is probably because it's quite distorted and it's difficult to recreate that in entirely. But what is clear is that the zebrafish, as well as other highly regenerative animals such as the salamanders and also frogs to some extent, have an enormous capability of repairing massive injuries to the brains, eyes, and spinal cord, both on tissue and on functional level. That's a kind of important driver for my research. And one of the secrets behind this is that they have a lot of stem cells in the tissue. And you can see there an animation rotating and a side view of an adult zebrafish brain. And those pink cells were basically, or pink staining there represents cells in cycle. So the fish received an EDU pulse, and it labels basically different active neural stem cell compartments in the animal. So they have a lot of neural stem cells, and these can also be engaged in repair. And that's very clearly seen in the panel to the right, where you can see an adult forebrain. The first panel shows an uninjured one, and then to the right, you can very clearly see an upregulation in the on the right hemisphere of the pink staining, illustrating and demonstrating basically stem cells reinitiating their cell cycle and contributing to basically the stem cell response for producing new neurons to repair the injured tissue. And this is one of the things that we are very interested in, of course, finding mechanisms how to engage and recruit neural stem cells. But one of the other things that has to do with their fast ability to functional repair is that there is also a question about the temporal issue of regeneration. And I will just mention what I mean with that. Because neurogenesis in general is a relatively long and complex process. So we may start from a dormant stem cell that's activated, for example, after an injury or a stressor, and then it goes through many different steps to differentiation, and also has to be maintained and integrated into the circuitry to actually play a major role. So this is, of course, in reality, what is important if we talk about really functional relevance of repair from newly produced cells. And in general, in any vertebrate, this is a process that takes quite a lot of time. In fish, that's about days to weeks. In humans, it's of course, in some cases, many months. So it's not a very fast mechanism to engage if you need to repair something rapidly or need to repair something essentially in an injured or diseased nervous system. And that is clearly a barrier and a problem in thinking about how to engage or use any type of stem or progenitor cells in neural repair. But what we thought, because the zebrafish can repair very fast, even if they take into the consideration the normal neurogenesis time, is that are there also a possibility that the zebrafish also have developed some specific tricks also to speed up basically their ability to regenerate, so they could speed up the process of differentiation or have other tricks to basically be able to functionally repair faster. So this is basically what we set out to study more deeply. And although we have done a lot of studies in the brain isn't the best system to study some forms of regeneration. That's why we turned into first the larval spinal cord in zebrafish. And the reason for that is that it's very easy to know if you lesion something. So we do a small microsurgery, for example, or laser ablation of the spinal cord in a 3-day-old larvae. At that point, the neural circuitry is very well developed. Most of the cell types that a vertebrate spinal cord has is already existing at that point. And also the spinal cord in general is very easy to compare, and it's very well conserved across vertebrates. And there you can see a clear lesion. And then the whole idea was that we use the larval system, basically do the injuries, then we can do different imaging and try to understand the process of regeneration better. And when we did that first, and here's some panels with different cells and how fast basically the larval spinal cord regenerate is, and we can focus there on the 1-day panel. You can 1-day post-lesion time point, and already at that point you can see a lot of cells and also fibers, or not a lot, but you can clearly see cells and fibers already spanning the lesion site. And at 2 days, there's actually a fair amount of cells already present really at the lesion site. And to the left, GFAP would be then different glia. It also trails in a little bit into the neural lineage because of the persistence of GFP. And then we have pan-neuronal markers in QC, and islet-1 GFP would be specifically secondary motor neurons and ventral interneurons. But very fast cellular regeneration is the key message of this slide. And the good thing is also that what we can do is not just look at the cells, we can also start looking at when they become functional at the lesion site itself. And we did that with GCAMP signaling, and I will play you one of the movies, and you can see there's spontaneous locomotion, and you see a burst of GCAMP. And then after 1 day after injury, you can also see that there's a signal, but it doesn't propagate through. And I also have to say that this is a great collaboration with Ethan Scott's lab and his excellent postdoc Giles that really helped us with this experiment. And when we do that and look at that, basically, we could find a time course when the spinal cord, in terms of neural activity, recover. And I will show you this summary of that because it describes it better and it's easier to see. So if we measure for just neural activity up and downstream, what one can see is in the uninjured ones, there's not a difference in the general GCAMP response. And then after the lesion, downstreams, of course, there's not much signaling going through. At that point, there's still 1 day after lesion, not much has happened. But you can already see that at 2 days, when we have also demonstrated there is a significant amount of cells and fibers, you can see some recovery in terms of neural activity, and basically almost back to normal at 3 days post-lesion. So a very fast recovery on in that way on GCAMP level, so indicating that cells are functional at that point. And then, of course, we can also correlate that to different behaviors. For example, swimming behaviors, the escape response, other types of swimming behaviors. Initially, after the injury, they can move around a little bit, but because the lesion is quite high or like low downstream, they still have a little bit of ability to move, but their ability to swim longer distances and respond is more or less also very close to normal at 2 days post-lesion. Also, we can measure basically force output and how that recovers after time, and that is also fast. They're also in line with the cells, the GCAMP, and the behavior that around 2 days they have recovered very well. So what we could say is at this point, although the repair, the tissue repair or the behaviors are not perfectly repaired, they already 2 days after a very massive spinal cord lesion, they have recovered fairly well in terms of cell and function. And then, of course, we wanted to look into that and understand how that works on cell level. And we first started looking at stem and progenitor cells and how they are engaged and activated. And what is important to notice and to know is that there's not a lot of ongoing neurogenesis or activity of stem cells. And that can be very clearly seen in the sham panel up there, where you can see proliferating cells in green, or for example, nesting expression also in green. And there's not a lot of cells that are labeled and are not at that time point. So basically, primary and secondary neurogenesis has already taken place. And as I said, the spinal cord circuit is very well actually established at that point. But what you can then see is that over the next 3 days, there's an activation of stem cells and also recruitment to the lesion site. And this is particularly seen then with the re-expression of nesting. But one of the things then that we noticed when we started actually trying to understand when exactly cells were produced by doing different EDU pulses to identify when different cohorts of cells were recruited or produced in the lesion, is that using either EDU pulses or also genetic lineage tracing, we could only label a fraction of the cells at the injury site itself at the early time points after lesion, basically indicating that even if we see a lot of actually regenerated or what appears to be regenerated neurons at the lesion sites, many of them did not seem to be derived from basically stem or progenitor cells. So there's a possibility that something else was going on. And then we started doing different time lapses to study that in more detail. And now you should observe in the time lapse that I'm just going to show you, there's some indicators in yellow and red, and that's basically the front of these transgenic lines that labels motor neurons and ventral interneurons. And this is now imaged just shortly after the injury. And then what you can see is it's quite a lot of time points in between. It's a time lapse over the 24 hours after injury. But what you can see is that cells gradually starts migrating in from both ends, essentially, into the injury sites. And we basically then also developed in toto cell tracking modules where we looked at regenerating cells in the spinal cord. And what we could see was that there was a significant recruitment of different, what appears to be neuronal cell types to the injury site. And that was a little bit puzzling. So then to test that cell proliferation isn't really impacting regeneration, what we then did was to then use different drugs to block cell proliferation. And we used a couple of different ones. And then in the panel, you can see there 2 days after lesion, you can see a lot of cells at the injury site, always kind of depicted with these boxes around it. But you see no difference between the drug and the DMSO treated, indicating basically that blocking cell proliferation does not impact early repair. Then we did the converse experiment, basically try to block migration. And first we used a more systemic and rough approach using nocodazole, that is a microtubule blocker. And when you put that at low concentration, it doesn't block cell cycle, but blocks basically formation of oriented microtubule cues, making the cells confused so they don't migrate correctly. And what you can see clearly from the panel in the upper left is that basically cells at the injury sites and also process is dramatically reduced or almost blocked when we do this. And we can also see that then that also impacts basically the different behaviors and also decamp active cells at the injury site. We then, to refine that approach, we used then photoactivable drug that also blocks microtubules called photostatin, and then we activated that specifically at the stumps to basically do the same, but without having the significant side effects that you can have from nocodazole treatment. And it also basically displayed the same thing, that if we block migration of these neurons coming in, we also block basically the repair. And to then get further into that, we had several different approaches, but I think the most telling one was to basically use genetic lineage tracing. And in this case, we created a transgenic line that expressed Cre ERT2, so tamoxifen inducible Cre in all neurons. And we then crossed that with one of the brain bow lines. We call them zebra bow in fish. And that cross we then called neurobow. So then we used that line and basically then genetically lineage traced all the neurons that were produced in the embryo, or we even did it just up to just before the injury itself. What we could see is that you can see in the control panel, you can see nice mosaic labeling, and I only show you the GFP channel. So the recombination is actually very good, but it is mosaic because I'm not displaying the other color hues in this case, but you can see nice labeling across the spinal cord. But already one day after the lesion, you can also very clearly see that a lot of these, so to say, pre-existing and also embryonically derived neurons and processes are also derived or recruited to the lesion site. And then a couple of days later, you can also see that they maintain and also build a significant part of the regenerated neurons at the very lesion, basically indicating that existing neurons are very important for driving the early phase of regeneration. And then what we wanted to do, and I'll see if I can, is to also combine that with the GCAMP and see if these early recruited cells actually also may be the ones that are pioneering the local circuit repair. So basically, they would be the first neurons that builds up the, so to say, functional scaffold of active cells in the spinal cord. And when we did that, we could actually see that these recruited ones, and there you see an arrow, and you can also see that that cell is also firing, and that also represents one of the early recruited neurons. And to really drill in on that and look at it in detail to separate what the contribution to the early functional recovery is from old, so basically cells producing the neurons versus new neurons defined as cells that are produced after the injury. We did different approaches, labeling approaches to label them in the embryo, or then we basically used EDU pulses that we administered directly after the injury. Combine that with the GCAMP, then correlated that to later fixed samples with antibodies, so we basically could identify where cells came from, if they were active, and also if they expressed different types of markers. We'll not go into great detail because it's quite complicated, but I will tell you basically what the outcome is, and that is that during the early phase of repair, so between 2 and 3 days after the injury, about 90% of the functional cells at the lesion size actually derived from the embryo, suggesting that they are really critical and pivotal for actually driving the initial circuit repair. While only 10% were new in terms of expressing post-pathetic neuronal markers, and even fewer of them, just a fraction of them, so just a couple of percent, 1 to 3% were actually active, suggesting that newly produced cells, they do arrive, but they're not very critical for the early phase and also for the early functional recovery in this case. We then looked and tried to identify what these cells that are recruited are, and again, I will not go into all the details. We don't see any indications that they would be derived from neural stem cells or progenitor cells from the different genetic lineage tracing, CDU, or the imaging. We also see that the cells don't, in a way, de-differentiate because they do not start expressing glial neural stem cell markers or proliferation markers. They always remain in the neural lineage. They express early post-mitotic markers, mainly of the ventral lineage, but we do see cells basically from across all the different lineages within the spinal cord. There's clearly a bias towards faster and more vigorous response for ventral lineages, such as the ventral interneurons and also motor neurons. And this, what we also can see by imaging them is also that they don't have a very elaborate morphology, so we don't think that very mature cells either so to say, retract and de-differentiate. It more looks like there's more of a kind of a dormant pool of neurons that can be engaged and used in this process. So basically, what we found and what we set up for is that we found one way for the fish how they actually initially can speed up the repair, and that is by basically engaging and using what we in a way could describe as dormant immature neurons, and they recruit them to the injury, and they are the ones that then rapidly form the new circuit and basically are the pioneers of functional circuitry in the spinal cord. And that's why we can say that there's two phases. There's a make-do and a make-new one. So the make-do one is what one would call from a regeneration biologist point of view and morpholactic, which is basically meaning tissue remodeling, is that the fish are able to recruit these immature neurons as fast responders to build up the first scaffold that then is driving the initial functional recovery. And this allows actually the animal in this time to buy time to regenerate to other means, basically to activate and engage the stem cells. And the stem cells then later indeed do build up the bulk of new or tissue that was lost. But this process is much later and takes place then over the next weeks in the fish. So it's basically by having these two overlapping processes, there's a fast track to make them cope initially, and then a second one that then gradually builds up and allows for more complete repair. And we also, and I don't have time to show you today this, is that there's also an actual connection with these things, because the recruited precursors also actually modulate and also guide the neural stem cell response and also shapes that one. I would also like to say in this case that it is that I'm not saying that the neural stems are not important and the progenitors that they are not important for the repair, because they are definitely required later on. Because if you do specific cell ablations in the spinal cord using a genetic system and a drug that basically allows us to, for example, ablate neural progenitors labeled with nestin, what you can see there in the middle is that if we just put the vehicle on, that you still have a lot of cells and red cells, that's basically neurons, while in the drug-treated ones, you see these large cavities and basically cells are lost. They don't really regenerate well, and the fish will not make it either. So it's also very clear. But the important thing is that we have these two different phases overlapping that orchestrate the overall repair process. And I'm just going to tell you a little bit more about how that could also be relevant for other animals. So first of all, in the spinal cord, also this type of kind of dormant or unusual, maybe immature cells are also being detected. For example, in rat, less so in mouse, but also, for example, in turtles, such cells have been found. And also known that they can be also recruited towards inflammation or injuries. However, it's very difficult to compare because a lot of the studies are a little bit incomplete in their experimental approach. So they are more hand waving than really demonstrating, but there's actually a possibility. But what is very clear is that such population for sure exists in the mammalian brain, in particular in ones with more elaborate structures and bigger cortexes. So for example, in sheep, a little bit in mouse, in the archaecortex. It's been shown basically there's some later population of cells that are produced in the embryo that differentiate very, very slowly over the time, but nobody has really looked into if they also can be engaged and also play some importance in repair later on. But it's at least very, I think, very thought-provoking and very interesting to think of that there could be dormant cell populations in the brains that could be utilized for brain repair. And with that, I would like to end, and I'm happy then to answer some questions later on. And of course, want to thank my lab, and especially these 3 excellent students, and also my collaborators and the funding agencies. Yeah, thank you very much.
Thank you so much, Jan, for an amazing talk. The imagery of your zebrafish model is amazing. Makes me want to convert animal models to zebrafish. So I'd like to open the session for questions, please. And I'd like to encourage any EMCRs or postdocs with questions to, of course, you can always type these in. You don't have to appear on video. Julian, you had a question. Would you like me to ask for you? So, Julian.
Okay, thanks. Thanks, Kelly. Jan, I really love such examples of showcasing the optical visibility and the live imaging capabilities of this animal model. I had a question about the repair process and what you identify as the old cells, the existing neurons, and what the microtubule disruption experiments reveal. Now, there are processes that are taking place. There's neuron outgrowth as well as somal translocation that is essential to the movement of the cells into the injury zone as part of the remodeling processes, if I understand this correctly. So do the microtubule processes, how do you see that process taking place? Is the microtubule organization essential for the existing cells to coordinate neuron outgrowth to form a bridge, so to speak, and then the latter phase is such that the existing neurostem cells are producing new neurons that then populate the area and reconstitute the local circuitry? Or is there a dual function for the existing neurons to not only build a bridge, but also to move their cell bodies in, because the positioning of those existing neurons into the site has an important function? And have you are you considering those areas of investigation to define? Yeah.
No, very good questions, and it's really something we haven't done enough on yet, and that I really would like a student to do so. I'm very, like, I couldn't convince any of my guys so far to do that detailed imaging, but that's definitely what we need to do, because there's a lot of interesting interactions, and we need basically really to, at very high resolution, look at the cells better to understand the process. And we can definitely see both really cell migration in that way that looks very similar to during development, that you have relatively elongated long cells that are kind of tangentially crawling in. But there's also clearly a component also of, so to say, neuron extension and so on. And I think that there are also different type, or clearly different types of cells that are involved in it that probably play their own part. But this is very interesting, and also how they also then interact with the other cells that are there. There's also, of course, the glia part of the central canal and the ependyma. They are slower, so actually the faster cells to react are neurons themselves. So, but this is really something that we absolutely have to look at more in detail. So
very interesting.
I agree with you. I'm trying to convince them that we do the imaging now. Yeah.
So I do apologize, but in the interest of time, we're going to have to move on. But thank you so much once again, Jan. And if anyone has any follow-up questions, we can pass those on for you. But thanks again, Jan, for a wonderful talk.
Yeah.
So next, I'd like to introduce our next speaker, which is